Okay, this is the last video for the last part of inheritance part 2. We'll be looking at the types of variation. Okay, so the variation that we are referring to here is the phenotypic variation, right? Variation in the expressed characteristics between individuals. Okay, so there are two types of variation that you'll be looking at: continuous and discontinuous variation. For the discontinuous variation, as the name implies, the classes of phenotype is distinct, discontinuous, no intermediates, right? Discontinuous. Okay, whereby for the continuous variation, there are no distinct classes, right? The phenotype has a complete, a continuous range between the two extremes. Okay, so for the discontinuous variation, the particular phenotype usually is controlled by very few genes, one, two, at most three, but most cases 1 or 2 genes. Okay, but for the continuous variation, right, the phenotype more often than not, right, is controlled by many genes, and we describe this as the additive effect of many genes because each gene with the different alleles, right, contribute a certain degree, a certain extent to the phenotype. Okay, so the final phenotype is the sum of the contributions by the different genes. Okay, and because many genes, right, so we give it a term called polygenic. So these are like polygenic phenotypes with the additive effect by many genes. Okay, environment usually play a part for continuous variation, although it is not a must, and usually does not play a part in discontinuous variation, but there are exceptions. Okay, so the environment part is not so conclusive, but usually don't have and usually have for the discontinuous and continuous variation. So some examples here for the discontinuous variation: we have the blood group, right? So you have ABO, AB. So either blood group A, blood group B, blood group AB, blood group O. There's no in between, right? No intermediates. And the fur color in the Himalayan rabbit as well, you either have the white color or the black color, right, in the different parts of the body, but you don't get like shades of black, for example, right? So distinct classes. And this is an example that's affected by the environment. If you recall, right, it's affected by temperature. Okay, so for the continuous variation examples: height and weight, skin color, blah blah blah, right? And eye color as well, right? So the different eye colors, and for each color there are different shades as well. So even for my eyes, which is like black, right, there are different shades of black as well, right? For those of us with black eye colors. Okay, right, and eye color is not affected by the environment. So this is an example of continuous variation that's not affected by the environment, right? So not a must. Right, so anyway, couple more examples, right? For the discontinuous, you have hairline. Okay, so either some of you, you can check yourself, right? Some of you might have the widow's peak, right? This particular protrusion of hair, right, on the forehead. Some of us don't have that. Okay, so you either have or don't have, right? So there's no in between. So this is two distinct classes, and the phenotype is monogenic, meaning it is coded for by one gene only. Okay, for the skin color, right, it is coded for by many genes, and it is also affected by the environment, right? How much sun or how much UV light one receives. Okay, then the hairline is not affected by the environment. Okay. So two examples. Okay. So anyway, a bit more discussion on what is the additive effect, right? So in this hypothetical case study, we have the phenotype controlled by two genes, and each gene with their different alleles, right, contribute different degrees or different extents, right, to the phenotype. Alright, so when the genotype is double homozygous dominant, the big G contributes 4 units to the phenotype and the big B contributes 2 units. So total there are the sum of the contribution of 12 units, right? So the sum of contribution, right? So therefore additive effect. Likewise for this other genotype here, okay? So if you look at the frequencies of the different genotypes, so assuming this is the cross between 2 double heterozygotes, right, you can draw the Punnett square and you can count the different genotypes, right? This would be the distribution, and the different genotypes would contribute different degrees to this particular phenotype, right? So you can actually plot the bar graph and it shows kind of like a range, right? So even though this is not even a polygenic example, right, because only 2 genes, right, it already kind of gives you a range, right? So you can imagine if this is a polygenic phenotype with many genes, then the range would be a bit more gradual, more continuous, right? So another example here with the color, in this case we have 3 genes, right? So each gene, depending on the alleles that they have, right, will contribute differently to the color phenotype, and the final phenotype, the final color, is the sum of the different contribution, right? So additive effect. Okay, so lastly, let's look at this particular example of a continuous variation in the phenotype of cork length in 2 different varieties of corn. All right, we have the Tom Thumb and the Black Mexican, where the Tom Thumb being the one with the smaller cork length and the Black Mexican with the longer cork length. All right, so the parents are both purebred. Okay, so we assume here we don't know how many genes are involved, right? But let's assume 5 genes, so that would be polygenic, right? So the parents, Tom Thumb and Black Mexican, they are purebred. Okay, so assuming the dominant alleles give more contribution to the cork length and the recessive allele give a smaller contribution, right? So the Tom Thumb with the smaller cork length would be homozygous for all 5, homozygous recessive, right, for all 5 genes, and the Black Mexican would be homozygous dominant for all 5 genes. Okay, so you see, right, they are both purebred, so therefore the individuals, right, have the same genotypes, but there is still a range of cork length, right, for the Tom Thumb and the Black Mexican. So this tells you, right, that these variation in the cork length, despite of the same genotype that the individuals have, must be due to the environment. Make sense? So the F1 is a cross between the 2 varieties, right? So the F1 would be heterozygous, right, for all 5 genes. So again, genetically the same, but has a range, right, in the phenotype of cork length. So again, the variation must be due to the environment. And then we have the F2, which is produced by selving of the F1, and you see a much larger range, right? The range is more extreme, right, compared to both parents and compared to the F1. That is due to genetic variation on top of the effect of the environment, right? So the parents and the F1 prove that environment play a part in cork length, right? So likewise, it should play a part for the variation for the F2 generation. But because the F2 is a result of selving of F1, right, which are heterozygotes for all 5 genes, so the F2 would have genetic variation. So genetic variation on top of the environment, it gives rise to a much larger or much more variation, and therefore the much larger base or the wider extremes that we can see in the graph. Okay, so this is an example of a case study showing you continuous variation, and by looking at the genotypes of the different generations as well as the presence of variation, we can deduce whether variation is due to the environment alone or due to both genetic variation and environment. Okay, so that would be the end of the lecture for inheritance 2.